The core goal of protein engineering is to screen stable and highly efficient proteins with designated functions from the infinite amino acid sequence space, or even create novel proteins that do not exist in nature. Proteins are composed of 20 types of amino acids. The total number of possible sequences far exceeds the total number of atoms in the universe, while only a tiny minority of sequences can fold correctly and exert biological functions. Directed evolution is the earliest practical protein engineering approach. It simulates natural mutation and selection, generates massive gene mutations artificially, and cultures and screens high-quality variants in batches. Comparable to blind shooting at a target in the dark, this foundational early tool of protein engineering requires screening hundreds of millions of samples, resulting in long R&D cycles and high costs. Intricate interaction networks exist among amino acid residues within proteins. Many performance-enhancing key mutations cannot be predicted in advance, which drastically reduces the efficiency of protein engineering. To break the efficiency bottleneck of directed evolution, rational design has become the second mainstream strategy for protein engineering. Based on atomic resolution protein structures resolved by cryo-electron microscopy and X-ray crystallography, researchers conduct site-specific modification on active regions to precisely optimize protein properties and realize targeted remodeling in protein engineering. Nevertheless, this strategy fails to resolve the core challenge of protein engineering. The complete set of rules governing the folding of amino acid sequences into intact proteins has not been fully deciphered. A large number of computationally optimized mutant proteins suffer from misfolding, precipitation, and functional inactivation after synthesis. Vast folding traps scatter across the sequence space, and errors accumulated from manual fine-tuning lead to repeated setbacks in rational design-based protein engineering. Whether through directed evolution or rational design, traditional protein engineering only performs minor modifications around natural proteins and hardly explores unexplored sequence space, which acts as a core barrier restricting the long-term development of protein engineering. AI protein language models fundamentally reshape the underlying logic of protein engineering. Pre-trained on billions of natural protein sequences, the models capture intrinsic rules of amino acid coevolution, folding compatibility, and functional sites. Free from natural template constraints, they generate de novo novel protein sequences with stable folding properties that have never occurred in nature. Matwing's Venus agent, developed by Matwing's technology, converts the capabilities of such AI models into an intuitive interactive tool, greatly simplifying the full R&D workflow of protein engineering. Researchers only need to input functional requirements, and Matwing's Venus agent can independently decompose tasks, generate candidate proteins in batches, and conduct multi-dimensional computational evaluation. It can also connect automated experiments to form an iterative closed loop. At present, only a small quantity of AI-generated candidate sequences need to be synthesized for validation in protein engineering research, delivering a remarkable improvement in R&D efficiency. Protein engineering has officially transitioned from modification of natural proteins to the stage of independent de novo creation of novel functional biomolecules. Current AI technologies still have limitations. They cannot fully reconstruct the dynamic and flexible conformations of constantly fluctuating proteins, and lack sufficient adaptability to complex intracellular environmental constraints including temperature, pH, and macromolecular crowding. These are urgent challenges to be addressed in modern protein engineering. However, technological iteration keeps accelerating. AI tools represented by Matwing's Venus agent are far more than auxiliary computing software. They serve as collaborative partners supporting protein engineering research on the sequence structure-function relationship. The ultimate vision of protein engineering is to decode the intrinsic language of proteins. Historically, protein engineering could only interpret the native protein codes evolved by nature. Empowered by AI technologies such as Matwing's Venus agent, humans will eventually rely on protein engineering to evolve from readers of biological sequences to co-writers of life's source codes.